Okay, continuing on with how cells move things across the plasma membrane, either into or out of the cell when they need to. I know some of this is probably maybe seeming kind of out there, kind of abstract right now, and we're just introducing these concepts for now so that we've heard about them, but believe it or not, they are going to come up as you move through your AMP courses. The cell membrane and moving things into and out of the cell winds up being extremely important for the physiological functions of your body systems. Okay, so I mentioned on the last video clip that the methods we talked about on the last video clip for transporting things were passive processes because no energy input was required. And as you see here on this side of our diagram here, everything you see there in that diagram, those solutes that are represented there, are moving from where they're more concentrated out here to where they're less concentrated inside the cell. And so if you have proteins that are helping that out, that's called facilitated diffusion because they're moving the way that they naturally would want to diffuse. And I don't want you guys to think that that only happens from outside to inside. Cells also move things from inside to outside when they need to because they have to maintain that stable internal environment. All right, active processes require energy input to force them to happen. And there are two types. One type is called active transport, and another type is called vesicular transport. Both types, though, use ATP fuel molecules to power that happening. Active transport is depicted over here on this side of the screen. That is where you have a membrane protein that is deliberately forcing some sort of solute to move against the way it wants to diffuse, against its concentration gradient. So if you notice here, these diamond-shaped solutes, they're more concentrated inside the cell than they are out here in the extracellular fluid. But you know, this is obviously some solute that the cell wants even more of inside the cell. Could be something like glucose, sugar molecules. Cells need those all the time for energy. So even though that cell already has lots of glucose in there, it's still going to want to capture more from the extracellular fluid and pump those on in. Now imagine if you just had a channel sitting here that would allow those diamond-shaped solutes to move across the cell membrane. Which way would they naturally want to go? They would naturally want to diffuse from inside, where they're more concentrated, toward the outside. So you can see why cells can't necessarily just open up random channels that allow things to freely flow from one side to the other, because that would not allow them to maintain a stable internal environment like they need to do. So active transport here is these solutes are being captured over here on this side, and then that carrier is opening up like a gate and allowing those solutes to move into the intracellular fluid, forcing them inside, and ATP is being used as a source of energy to make that happen. So that's active transport. Vesicular transport, what in the world does that mean? What does vesicular mean? Sometimes cells have to package things in a membrane covered little compartment or bubble to bring them into the cell, or to transport things to the cell surface so that they can be released to the outside of the cell. And those little membrane covered packages or bubbles are called vesicles. If a cell is using vesicles to package things that are outside, out here in your extracellular fluid or ECF, and bring them inside, that's called endocytosis. Endo, like inside, you're bringing something inside the cell. If the reverse was happening, it would be called exocytosis. So cells, let's say a cell is producing some kind of protein hormone, and it needs to secrete that hormone to the outside of the cell so it can do its job. It'll do that by packaging that hormone up in a little membrane-covered vesicle, and it will, when needed, it will send that vesicle up to the surface of the cell, and that hormone will be released. So that would be called exocytosis. cytosis, when a cell uses these little vesicles to secrete or release substances that they've made. All right, so with endocytosis, there are 3 types depicted here. We're not going to get too heavy duty into this. You'll hear about these on occasion in biology. You'll especially hear about the first one over here, phagocytosis, P-H-A-G-O, cytosis. So P-H-A-G refers to eating, so phagocytosis can literally mean cell eating. And this is a process that your white blood cells use, for example, when you have white blood cells that act as little warriors, and they're supposed to capture things like bacteria and viruses and bring them inside, and then they destroy them. Once they've brought them inside, they kill them and digest them. That's what's being depicted over here. This is actually supposed to be a bacterial cell, or a bacterium, and it is being captured at the surface of this cell, and a portion of the plasma membrane here pinches off, surrounds it, and brings a little membrane covered bubble inside the cell. That's called phagocytosis. And now you've got that little bacterium there inside, and it's going to wind up being killed and digested. That's phagocytosis, or cell eating. Pinocytosis, depicted here. Your cells do randomly pinch off portions of their plasma membrane and bring those little pinched off portions inside as vesicles. And those vesicles will contain solutes that were floating around in the extracellular fluid. So they kind of randomly sample their environments. That's called pinocytosis, or cell drinking. And the third type depicted over here, remember we were talking about membrane receptors that will, you may have a chemical messenger sitting here in the extracellular fluid, and it docks onto a very specific receptor. They come together like a lock and key fit, and sometimes when that happens, that triggers receptor-mediated endocytosis, where only when that happens, that receptor binds that substance, whatever it is, then the vesicle pinches off at the surface and brings whatever it is that had docked there on this plasma membrane to the internal part of the cell. So that's called receptor-mediated endocytosis, because the endocytosis doesn't happen until that substance binds to its receptor. All right, so those were our examples of active processes that cells use to pump things into or out of the cell, or deliberately force them in or out. The next lecture for Chapter 3 will be about osmosis. We said osmosis is a term used to describe the diffusion of water molecules across a selectively permeable membrane like your cell membranes. And it's such an important topic for physiology that we're going to have a completely separate lecture video clip about osmosis.